the SRF for coming here and tell you about the work we have been carrying out in the past and present and future, possibly thanks to the ESRF and to what it really did for structural biology in Europe and worldwide. I'm really, really honored to be here. And I will start without wasting other time of thanks, but because I have been a user of ESRF throughout times. There are few beamlines that deal with structural biology that I didn't use. So I'm an enthusiastic user and fan of this amazing scientific facility. Anyway, what am I going to talk to you about? I'm talking to you about ferritin and about how it interacts with this receptor and about how we, as human beings in biotechnology and nature, namely pathogens, have exploited this interaction in the past and how we hope to exploit it in order to, for example, deliver drugs or carry out more efficient diagnosis and of pathologies such as cancer. Now then, how am I going to structure this talk? We are going to talk about ferritin as a selective nanocarrier, how its receptor, because ferritin is not only residing into the cells, but it's also circulating in the bloodstream, it interacts with the receptor to assist transferrin in uptake by cells, how we did determine the structure of the complex between ferritin and the receptor CD71, the features of the structures, and then some conclusions. Oh, brilliant. Now then, first of all, let's talk about ferritin, which is a very familiar molecule to many of you, I imagine. It is a sphere, which is made up of 24 subunits, which are tiny proteins that are formed up by 4 helices, main helices, and loop. It has the main function is storing iron storage within the cells and oxidizing iron. So it has also ferroxidase activity. And why is that? You, of course, need iron, fundamental, but iron is also quite active in redox reactions. So you want to store it in a safe way in the cell. You cannot live without it, but if it's free in the cell, it's extremely toxic because of several production of radicals due to its extreme activity as a redox center. Outside the cell, ferritin also works as an iron carrier. And as I said before, it's made up of 24 subunits. The total weight is 500 kilodaltons. And the size, however, of the particle is much larger than what we would expect given its molecular weight, because it's an empty sphere. And the external radius is about 12 nanometers, and the internal radius is 8 nanometers. So what is the interest? I mean, it is per se a nanoparticle. It has the size, the symmetry. It is definitely the ideal nanoparticle. And the advantage of it is that it is completely compatible with physiology. It resides in the bloodstream, it resides in the cells, and it can pass the body barriers, including the one between the brain and the circulating system. And what about biotechnology? Well, of course, if you use something like that, you can use it to encapsulate something. This is the ideal object you can imagine to use for encapsulation. However, encapsulation is sort of
Bravo, well done.
okay.
Allez, laisse tomber, c'est trop compliqué.
No. Perfect. So it is really ideal. However, the procedure that is well defined for the encapsulation is a bit harsh. So what you do, you have your empty sphere, you expose it to quite extreme pH for a protein, either very low or rather high, 2.5 or 10.5. This disassembles the sphere. And if you do that in an environment where you have, for example, a drug, and then you reassemble it, and then you eliminate what was in the external part, you can have an encapsulated compound, a drug, for example. What is what can I say, drawback of this procedure is that proteins are sort of delicate objects, and they don't really like to be exposed to acidic or to alkaline pH. And once you have encapsulated it or before, there is another advantage that it's a protein, so you can modify it a number of ways, including simply cloning it and adding different tags that can add specificity, or you can link them covalently via the sulfide bonds. You can use a chemical procedure. So there is a almost infinite way of adding tags and other molecules that can target precisely the molecule to any receptor or cell population. But actually, there is not the need for that, at least if you want to target the cancer cells, because actually ferritin, as we will see, is already targeted because it recognizes a receptor on the cell that is the transferrin 1 receptor. What is the transferrin 1 receptor? It is a well-behaved receptor, so it has a transmembrane segment. It is a dimer. We have an interface, an helical domain which mediates the dimerization. We have two domains, the so-called apical domain and the protease-like domain, here represented respectively in yellow, orange, and red. And how does it work? It resides on the surface of the cell, and it recognizes transferrin, which is the classical iron transporter in mammalians. And then once the transferrin loaded with iron is bound, you have an invagination in the cell, the formation of an endosome. The pH of the endosome becomes more acidic, iron is released and either stored inside ferritin, or, for example, inserted in the heme and then going to whole stand of different iron proteins, and then the receptor is recycled. This is a very classical mechanism for uptake and then release of a receptor. And it is very important that iron homeostasis is regulated, because if you have too little of it, of course, iron deficiency, number of pathologies, but actually you can have too much of a good thing. So iron overload is also related to a number of pathologies, because if it is no longer safely incorporated in ferritin and is free in the cell, either per se or incorporated into heme, you have inflammation, cardiovascular disease. It can be conducive to DNA damage and cancer, and it is also interestingly related to neurodegenerative diseases. So you can have too much of a good thing, namely iron. Our interest was both in the use of ferritin as a nanoparticle, both for diagnostics and therapy, and in the way it recognizes this receptor, because you may want to, for example, as we will see, target other ferritins to this receptor. And why do you want to target the transferrin receptor? It is the physiological one, but more relevantly, it is already a way of targeting cancer cells. So as shown in these slides, a huge number of cell lines, malignant cell lines, as you can see, this is a staining for the CD71, the transferrin receptor. In normal breast, normal lung, the staining is very poor. However, when you have cancer cell lines they vigorously stain for the CD71 receptor. This is because cancer cells are very avid of iron. They replicate so quickly. They need a number of nutrients, a number of compounds, iron included, to sustain their incredibly fast replication. So we can see here that a number of cancer lines, including, for example, some that are difficult to target, such as the pancreas cancer, are already hyperexpressing the CD71 receptor. In this way, if you target the receptor, you are targeting cancer cells without having the need to engineer your nanoparticle. And so we wanted to understand this interaction, both because we want to understand how life works, but also because it could be exploited to better target any nanoparticle, including ferritin, modified or not, to cancer cells. And the classic thing one would do, we are structural biologists, we like to go to synchrotrons, was to try crystallization. However, crystallization was, I would say, very difficult, if not impossible, because since we have 24 potential binding sites on ferritin, in principle, because we didn't know how ferritin bound, but I mean, one would assume via just one molecule, but we have a dimeric receptor, we had aggregation, we had the formation of large aggregates because there was a bridging between one ferritin and two or more receptors, and precipitation. The system is heterogeneously, intrinsically non-homogeneous, and that, as everybody knows, is not conducive to crystallization. So we decided to try to do cryo-EM. And actually we used a very simple things work. That was just to bind the receptor to the two beads via tag, via nickel binding tag, and then to add ferritin on the column, and then to detach this from the column. In such a way, we had at least monomers. We didn't have the cross-linking problem. We didn't have the problem of large particles, heterogeneous ones. Simple works, at least in biochemistry. And then I'm here I give a very brief scheme of the flow of cryo-EM experiments. So you have to basically freeze take a tiny amount, 20 microliters or so, of your sample, layer it on a grid, and then you have to eliminate the excess liquid. You very quickly dip it into liquid ethane, plunge freezing. So you lay, you blot, you freeze, and then you have a very thin film in the holes of your grid, and then you chuck them into a cryo-EM, a microscope, and you you start to collect data. And of course, the vision of ESRF has been ample in this respect. As you know, there is a beamline in the ESRF where you can also do cryo-EM. So you can really put in synergy the several methods of structural biology. So actually the hole are the tiny hole of your film on the metal grid, copper, gold. I mean, there is only fantasy is a limit for that. And then you collect your data on the microscope, and you have all the different projections on your image. However, the problem is, of course, signal to noise. That is this is an exceptionally good case. The particles the image don't have a lot of contrast because this is a protein in water, so the material is not so different. And so you have to enhance them. And you enhance them by simply summing them and then dividing. So you enhance your signal. This is the first process, which is called 2D classification. And then when you have all the different projections, you can reconstruct the structure or the structures, because this is one of the beauties or the curses of the method. you 3D classify, and then you refine, which is a different refinement with respect to the classical crystallography one, because you never change your data, no way. And then you get your structure or your structures. So this is the first data we collected in the US, and these are the better data we collected at the SRF. As you can see, in this case, the particles are visible in an exceptionally good way. Normally, you don't really see them. You try to imagine them. But with ferritin, actually, you can almost always see them. Ferritin is the myoglobin of cryo-EM. And then you see your beautiful 2D classes, and you're very happy because you're directly seeing alpha helices and whatever. So it's moving as an image for me. And then you look for the classes that, 3D classes that make sense, and you get intermediate maps, and then you refine. And at the end of the day, you get your structure. And I would like to, where is it? No, not there. Show you how that in the structure we really have the two projections. One is the side projection, this one, and one is the top projection. So one is the little butterfly, which has lower electron density, and the other one is the 90 degrees rotation, that is the top view. Don't be, what can I say, deceived by the fact that these are two large proteins. This interface is quite large. It's about 1,000 square angstrom. So it's a good, well-behaved protein-protein interaction with high affinity. The dissociation constant is in the nanomolar range. Okay, final resolution, I mean, for a protein crystallographer is not so exciting, but the resolution in this region is much higher. So what do we see at the end of the day? And is it relevant? Now then, what we see is that the contact between the two proteins, as expected, there are, of course, two binding sites on the receptor dimer. And we have that in the receptor, the binding region is made up by 3 beta strand and 1 alpha helix, beta sheet 2, strand 8, 1, and 2, alpha helix 2, and on ferritin we have 2 helices and 1 loop involved in the contact. And in the receptor, the only region that is involved in this binding is the so-called apical domain. So this top tiny domain, which actually is not involved in the binding both of transferrin. This is the complex between transferrin and the CD71 receptor. As you can see, transferrin is the orange blob. It really has a very extended contact with the protease-like domain. This is another protein. This is called HFE. It is the protein involved in a pathology called hereditary heterochromatosis. And actually, very interesting, this protein decreases the affinity of the receptor for transferrin. So if it doesn't work properly, if this complex is somewhat disrupted by mutations, your uptake of iron is too high, and you have pathologies due to the fact that you have an excess iron in your cells. And as far as the so-called apical domain, it didn't seem to have any physiological use. Actually, it's involved in the formation of complexes which are not really something you want to have. That is, complexes with viruses. This is the Machupo virus, coronavirus, that, I mean, as we sadly know, can cross species barriers, and is one of the candidates, for the future pandemic. And it is even recognized partially by Plasmodium vivax, the receptor binding protein of Plasmodium vivax. So this receptor is also hooked by a number of pathogens that want to enter cells. So it's used by the good guys, but also by the bad guys. And it targets this region. And one would ask, why do we retain this region? We didn't know before, because it was very well known that this is the surface exploited for binding of transferrin. No use was assigned to this domain that seemed to be there only to be a door for the bad guys. Now we realized that this was used for the internalization of ferritin. And so this explained two things. One, the fact that the binding of transferrin and ferritin is independent on the receptor. So you can bind at the same time transferrin and ferritin. There is no exclusion of the two. And second, this domain that seemed to be there just to let the pathogens in is actually a physiological use. So this was our first observation. And more pathogens, for example, the parvoviruses, this is the huge virus, bind also to the apical domain of CD71 receptor. And as you can see, it is the same region or very similar region exploited by human ferritin. So this made sense as far as the binding with pathogens is concerned. Why is this region conserved? So for parvoviruses, the structure has a very low resolution. However, the structure between ferritin and the glycoprotein 1 of the machupovirus, which infects rodents, but can also infect humans, luckily in a very small number of cases. There is always a mutation awaiting. And so we inspected the residues that are involved for binding ferritin and the residues that are involved in binding the virus. So what we observed is that there are some residues, the blue ones, that are used only by ferritin, some residues, the yellow ones, that are used only by the virus, but the central part of the interface is crucially exploited both by ferritin and viruses. And actually, it is these amino acids that actually lead to jumping this changing specificity when these viruses affect the different species. So this makes sense. So the viruses change their host specificity when they change the residues that recognize this region. And so we're right now the affinity is not so high, although actually a protein structure could be solved, but there is always the possibility that other viruses of this family change their specificity and can bind the CD71 receptor, therefore being potentially capable of affect humans, because the receptors can bind to this receptor, their external proteins, and therefore being internalized. So this is a key region to monitor in order to understand whether mutation in viruses can help viruses, say, that affect hamsters to infecting humans. And these are the residues that on the CD71, the common contacts between ferritin and the machupovirus that are central to the domain. And in the center of the domain, this is also the comparison between the human ferritin complex, machupovirus complex, and receptor binding protein of Plasmodium vivax. Again, the common contacts, these are always in the center of the receptor, and are the ones that are exploited for the recognition, both in viruses and also Plasmodium. So this is really a crucial region for affinity and specificity. Another thing we wanted to do, of course, was to validate our structure, because even when you see a complex on which, of course, we totally believe as structural biologists, you have to convince those nasty creatures that are referees, who will tell you, yeah, you see that, but are you sure that this is the real complex? And you have to work hard to convince other people that your beautiful structure makes sense in biology. And so we were focused on ferritin for reasons that I will tell you later. So we identified what we believe are the crucial determinants for binding. That are 3 residues on the one of the alpha helices, helix A, and 2 residues on the external loop of ferritin. Since it is between helices B and C, it's called the BC loop. So we carefully checked the context, and we identified these 3 residues: Q, glutamine 14, D, aspartic 15, arginine 22 on the helix, and phenylalanine 81, glutamine 83 on the BC loop, and then we put the 5 mutations together in a mutant that contained overall 5 differences. And why did we choose these residues? First, because their interactions are convincing, because you have an interaction at longer distance, something that is not the fundamental one. These are very sound, well-behaved contacts. And the other, because these residues are absent in ferritins that don't bind to the receptor. Something I forgot to tell you is that naturally occurring ferritin in our bloodstream is made up by 2 chains, the so-called heavy and light chain. Only the heavy chain binds to the receptor, the light chain doesn't. So simply by comparing the sequences of the 2 heavy and light ferritin, we found that differences that actually occur in the binding site. And is also, we also checked other ferritins, for example, ferritins from other species, because ferritin is everywhere, including bacteria, including archaeal bacteria. So it is really a very successful machine in life. Really, you want to keep iron under control, but you can't live without it. Now then, so with 3 mutations on the alpha helix, 2 mutations in the BC loop, we were very conservative. We mutated the glutamine, aspartate, and arginine into alanines, as well as the phenylalanine and glutamine. And we carried out a very simple experiment with surface plasmon resonance to assess the binding and affinity dissociation constant. And this is the wild type, 10 nanomolar KD, very rather tight contact affinity. And then mutant A, the one in which we change the residues in the alpha helix, we have a substantially decreased affinity. Mutant B, the one on the BC loop, even lower, although at very low affinity, the numbers you get from the instrument are really not reliable. So we can just say that they are at least 100 times lower. And then in the quintuple mutant, in which we had all 5 mutations, basically no binding was detectable. So we were convinced that actually we had something that really was the surface, at least the surface that allows measurement in vitro, because this is made with isolated proteins, of course. And then we, since we convinced ourselves, we didn't consider ourselves enough, convinced ourselves enough, we did go for cells. So we, this is not very visible, I do apologize. So we attached a fluorophore to our ferritin, and we observed that the wild type, of course, was internalized by HeLa cells, and that the 2 mutants had marginal, if any, uptake by HeLa cells. And these are actually the numbers determined by cell sorter. So not only the binding is affected in vitro, but also in vivo. Of course, the referee wasn't convinced, and so he asked also to carry out a gain-of-function mutant, which actually is much more convincing. And so we took the light chain of ferritin, where binding doesn't occur, and we introduced the mutations that we believe are the positions that we believe are relevant, this threonine, this glutamine, this arginine, and phenylalanine, because the glutamine is already there. And indeed, this mutant, this is the binding for the wild-type heavy chains. This is the binding non-detectable for the wild-type light chains. And our mutant has a binding of the same order of magnitude as the heavy chains. So not only these positions in the protein chain do eliminate binding if deleted, if changed to simple alanines, but introducing these side chains in a ferritin that wouldn't normally bind the receptor actually do induce very convincing binding. So we were very happy, and we could publish. Now then, as I mentioned, what we want to do is, of course, visualizing the interface. And by doing so, we also managed to say something about how pathogens exploit this receptor for entering cells, but also why the hosts, the victims of the infection, cannot defend themselves so well. Because in the interaction between parasite and host, or pathogen and host, there is a so-called phenomenon called race to the arms. That is, the host doesn't want to be infected, and so in the process of natural selection, individuals that carry modified targets do acquire fitness, survive better, and they are selected. This is called natural selection, actually. However, if the structure that is the target of the pathogen is important for physiology, you cannot change it so much. So if you have a structure that is used by the pathogen that you can change at will, or that on which selection can work at will, you have the race to arms. The host has a mutation and the pathogen has a mutation, and so on and so forth. And you have parallel evolution of the specific protein. However, if the structure that is targeted by the pathogen cannot change so much because it is relevant for a physiological function, you cannot mutate it. You are restrained only in those mutations which are neutral in function. So by using part of the receptor that is also used for the uptake of ferritin, the pathogens have the advantage of the fact that the host cannot mutate, okay? Now then, the last part, the last thing I want to talk to you about is how we exploited the knowledge we acquired with this structure in order to engineer a better nanoparticle for drug delivery and possibly diagnostics. That is, we took this archaeal ferritin, actually the structure of this protein was determined at the SRF, which is different from the human one. It has this huge window, trigonal window, and it has also an advantage. That is, it doesn't need harsh pH to be disassembled, but it disassembles in a very mild way simply when you have a low concentration of cations at low ionic strength. So we can disassemble that and reassemble that in a way that really increases the yield of loaded nanoparticles, and it doesn't actually damage the cargo. The problem is that this Archaeoglobus fulgidus ferritin have a BC loop which is different from the heavy human ferritin loop, and it doesn't bind at all to the CD71 receptor. So you can load it with cargo, but then it's not going to be uptaken. So what we did, we simply carved the original BC loop and substituted it with the loop that we have in the human heavy ferritin chain. And we produced a chimera, a humanized archaeoglobus, archaeal ferritin with the human BC loop. And to our joy, if not amazement, actually the humanized archaeoglobus fulgidus ferritin is uptaken by cells. See that the green fluorescence gets inside the cells. And as we solved the structure, we observed that the structure of the loop, this is the humanized ferritin, this is the human heavy ferritin, this is the original molecule. The structure is extremely similar, I would say identical in the loop. These are the changes we introduced to mimic the human ferritin on this loop. And this is actually the overimposed structure of the original ferritin and of the humanized one. So you may see, for example, here we have a tyrosine, here we have an arginine. So the key residues are there and they are in the same position. So it potentially binds, and of course we are now producing the complex between the engineered ferritin and the receptor to prove again that the structure are the same, and of course to see if there are other changes that we may introduce in order to improve binding, which can be much more easily deduced if you have the actual complex rather than a simulated one. And so to conclude, we characterize the complex of human ferritin heavy chain with the transferrin receptor 1 by single particle cryo-EM. We explained why the binding of ferritin to the receptor is independent from the binding of transferrin. We gave an explanation of why the binding to this portion of the receptor is advantageous for pathogens to enter cells because the host cannot defend itself by mutating it. And last but not least, that we can transplant this loop to induce binding to the receptor for other ferritins, thus paving the way to using ferritin as a totally biocompatible nanoparticle. And of course, I would like to thank people involved in this study, myself and Alberto Boffi and Martina Piret, the Department of Biochemistry, Claudia Testi and Paola Baiocco at the Italian Institute of Technology, Amedee at CUNY. We really have to thank Amedee because thanks to him and to his support, we could import cryo-EM in our group. Of course, the Institute for Molecular Biology and Pathology of the CNR, the Catholic University of Rome, and Filippo Mancia at Columbia University, ESRF and CM01 for allowing us to carry out the experiment, and the Italian Association of Crystallography and the Pasteur Bolognetti Institute for support to with grants to travel and carrying out the experiments. And of course, thanks to you for your attention, and thanks to the user organization for inviting me.